Hello, everyone. I'm Dr. Andrew Kaufman, and this is a Medicamentum Authentica special presentation. I'm very excited to discuss this video and paper with you, and let me tell you how I came upon this material. I was recently on an interview on the podcast Anarchast to talk about my upcoming trip to Mexico, and Patrick Smith mentioned that he saw a video of a virus attacking a cell and destroying it. And of course, I was very skeptical of this because, as you may well know, there are no microscope technologies available that can visualize something as small as an alleged virus particle, which is on the order of around 50 to 100 nanometers, while it's still alive. And electron microscopy is the only technology we have which can visualize things that small. And in order to take biological cells or samples of tissues and particles and look at them under electron microscopy, you first have to essentially kill the tissue, destroy it, and completely replace all the biological materials with metals, because certain metals stains are the only thing that you can actually visualize on an electron microscope. So I was a little bit skeptical, as I said, and then I wanted to learn more. So I asked Patrick if he could send me the video or a paper wherever he saw it, but I don't think he was able to locate it. So fortunately, a friend of mine sent me a link to the video, and I think this is what he's referring to. So let me get right into it so I can help you understand what is going on here in this video and the corresponding scientific paper, which I will go through with you and explain. And my aim is that we're going to watch the video one time and without any real discussion of what's going on, so you can get a first impression. Because many people who have seen this video feel that it shows, a virus attacking and destroying cells. So I'll let you be the judge. Then I will go through the paper, which describes the whole process of how this video was created. And then I'll show you the video again, and I'll help explain what's actually going on. So this is the video, and it's from researchers at Duke University, my alma mater. So let's take a look. All right, I'm going to briefly stop the share for a moment so I can go over that. So you see there's a lot of production value there because we have the dramatic music and we have all of the overlaid text, which uses the word virus over and over again. And even the title of the video, sorry, I'm blanking, is called How to Catch a Virus. And this is on Duke University's YouTube channel, and at present it has, 93,000 views. So not exactly a quote-unquote viral video, but still definitely seen more than most of my material in general. So let's now look a little closer, because in the description or the show notes from that video, they reference a paper. So I will share my screen again now, and we can take a look at this paper. Okay, so here is the paper, and it's from Nature Methods. So interesting, not from the main Nature journal, but Nature Methods, where there are a lot of publications about new methods that you can use for scientific study. And I think that's really the purpose of this paper, but we'll get to that. So it's called Capturing the Start Point of the Virus Cell Interaction with High-Speed 3D Single Virus Tracking. So there are some problems here right off the bat because one of the things that it uses the term virus, cell interaction, and single virus in the title. However, there's actually no viruses in the paper. So let me go a little further and I'll explain. Now, really, in order to understand and debunk a paper like this, or this particular paper, we can really just look at the abstract, and that tells us enough information to know we can discount the conclusions of this paper, at least how they're presented in the abstract or in the title or in the video. However, I'm going to go through a little more detail with you. But before we get into the text of the paper, let's just talk about a couple of things. So one thing is that this paper is written in such a way that there's the introduction, the results, and then the discussion, and then the references. So you could look at this paper and read the complete paper, what you think is the complete paper, but it doesn't include the methods section, which is how they do the experiment. Now, it does describe some of this in the results section, interestingly, which is not results, obviously. But I think that they don't really want you to read the methods section, and they do this a lot in papers in the modern era, but this was always the first part of the paper that you read, according to my classical scientific training. The other thing of note, which is very interesting here, is if we look at the very bottom of this page, you see that the authors are from the Department of Chemistry at Duke University. So they are not from biology, they are not virologists who study biological phenomenon. They're chemists who study chemical phenomenon. So it's kind of interesting that they would be doing this type of experiment. So now we can look at the abstract here a little more closely, and I've highlighted a couple of key portions. So the first highlight says an active feedback single particle tracking method. Okay, let's just dissect what that means. So as I explained earlier, there's no microscope that can visualize things the size of alleged or so-called viruses or viral particles while they are intact or biologically viable, if that's appropriate. So what they're using here is particle tracking method. So in other words, they're putting some kind of homing device, attaching it to the particle that they're looking at, and I'll get into what that is very shortly. And they're using the tracking data. Okay, in this case, they use lasers and a fluorescent protein, but this is very similar to, using GPS on your phone, and you know that there is an error. So in other words, they might be able to pinpoint you, within 10 feet or within 3 feet, but not the exact location like when we visualize something. So there's an error there, and there's a deception that we're not actually looking at any, virus particles, but we are actually tracking them using technology, okay? Now, the second part of this highlight says, with simultaneous volumetric imaging of the live cell environment called 3D TRIM. Now, so there is some microscopic imaging, but if it's not of the small particles, it's of larger cells in a cell culture that they're using. And if you look down at the next highlight, there's a simple monolayer culture. And I'll tell you a little bit more about that as we discuss the methods. But that is being visualized under the microscope, but there's also computer modifications to those images, as you can see in the video. Okay, so we have the particles being tracked by some kind of homing technology, and the artificial cell culture in a monolayer, right? So in other words, a single layer of cells, not like we have in our body where there are many layers of cells in the tissues organized in some kind of three-dimensional structure. And those are imaged directly. Now, in the final sentence, I highlight biological systems because they say this presents opportunities for capturing fast three-dimensional processes in biological systems. And they're talking about their technology of using this tracking method and this other direct visualization method. But in this particular experiment, they did not use biological systems. And that's a big part of the problem. What they used is artificially created systems that have biological constituents, but they're not biological. In other words, nothing in this experiment resembles what we would find in nature or consists of what we would find in nature. So I'm going to go now a little bit further in the paper to the results section where it actually describes some of the methods. But I thought that it was written more simply in this section than in the methods section, so I use this for teaching purposes. So in the left column, we can see it states in the highlighted portion, briefly, a rapid 3D laser scan is generated by an electro-optic deflector and tunable acoustic gradient lens. The scan laser spot excites photons from the fluorescently labeled diffusing viral particle, and photons are collected on a single photon counting avalanche photodiode, which also acts as a pinhole to provide confocal detection and prevent detection of out-of-focus virions. And that last part about the confocal detection in the pinhole allows it to have essentially depth of field, so it's not confused about the distance away from the sensor as much and can follow the particle over time. And then the photon arrival times are used to calculate the real-time position. Now that's a key sentence because they're used to calculate the real-time position. In other words, they can't measure the real-time position directly. So if we were able to, for example, have a magical microscope that could see those tiny particles, we could have a clock, right, superimposed on the screen of what we're looking at, and we could see the exact location at any given point in time. But this has to calculate the location, so that is a source of error. Okay, so essentially what this does is that there are these particles that are used in the experiment, which I haven't described yet, but they have a fluorescent protein label on them, and they use a laser to essentially excite that fluorescent protein so that it emits fluorescence in the form of photons as described here. And then those are detected by some sensors. And based on, the delay between the firing of the laser and when the sensors detect the photons back, that's used to calculate the location of the particle at any point in time. And so this is the technology that they did this. And then in the next, so what they did is they superimposed this data over the visualization of the cell culture in the background. And that's what you see in the video that flips upside down in the middle of the video. Now, the next highlighted portion says, for this pioneering study into the early stages of the virus-cell interaction, we investigated the journey of vesicular stomatitis virus, or VSV-G, pseudotyped lentiviruses from the extracellular solution to the cell surface and beyond. This specific viral-like particle, now that's the key language. Okay, so they didn't go into nature or into any infected host with a viral illness and pull viral particles out of nature in a purified form. Instead, they made a viral-like particle, and they chose a specific genetic makeup because they say that this particular viral particle, which is completely laboratory man-made, has a tropism, which means that it infects different cell types. Okay, so it's sort of convenient in a sense, I suppose, from their point of view. And then they said it's been established as a standard for gene delivery. Interesting. So sounds like the technology that is in the injections for COVID, right? That it's essentially a gene delivery system, and this is a synthetic kind of particle made for gene delivery, just like in those injectable products. We utilized an efficient and well-characterized internal virus labeling approach whereby fluorescent proteins fused to the HIV-1 viral protein R are packaged into budding lentiviruses. So in other words, they took genes from HIV and mixed them in there with the genes from the VSV-G in order to make these pseudoviruses or viral-like particles, which aren't found in nature and aren't actual viruses, which, as you know, don't exist. Now, the next highlighted portion actually gets into some of the results. So remember, what they're doing is they're tracking these synthetic particles that they made through space using a homing device, and they're doing this right on top of a cell culture, which they can see under the microscope and image using various techniques directly. So here are some results in the highlighted portion on the left. The virion closely approaches and intermittently touches the surface of several cells over the course of the trajectory. So essentially, their tracking data shows this particle like, moving around, and occasionally it bumps into a cell. That's what they're saying here. Now, we know that in the next highlight, they say this value of 0.5 micrometers was used as a threshold value for determining contact. So they aren't really able to tell that there's actually direct contact between the particle and the cell. They make an assumption that if they measure the distance and it's within 0.5 micrometers, which are millionths of a meter, that they're saying that counts as actual coming into contact. So they don't even really know if it actually bumps into, but it at least comes very close to, depending on the error of their tracking techniques. And now what's the significance of this? Because, okay, we dropped a particle into a cell culture and then it moves around and eventually it bumps into something. Not really terribly surprising or informative finding. Now, what they noticed is that when these particles seem to bump into a cell, sometimes they left like they bumped and then they moved right away, and sometimes they stayed a little bit longer. And they tried to look at those interactions. However, what they did not see is that they never saw the particle enter a cell. They never saw any cells be destroyed or changed in any way. Okay, and they wrote right here in the highlighted portion, it can be concluded that these initial interactions are not related to receptor priming, but potentially related to cell morphology. And what that means is that there's no evidence that these particles actually had a receptor interaction. In other words, they didn't have a chemical interaction with the cells. And this is really important because we are told that the whole way viruses work, and the virus tropism is related to this, is that the virus has a protein on its surface that the cells that infect have a receptor for, okay? But in this experiment, they couldn't observe any data that told them anything had to do with receptors. And the way that they reasoned this out is because they had different cell cultures, and some cell cultures supposedly have more receptors that this virus will bind to, but they found no difference in these bumping into interactions between those different cell types. Now, what they're saying here about morphology, which is the shape of the cells, is pretty simple, that if the cell had an irregular shape, like there was something like a phylopod sticking out of it, which we see in various cells, that could sort of, capture or bump the particle more likely. And so depending on the shape of the cell surface, it may be more likely for a particle to bump into it. That's basically what they're saying. So not really a very informative observation. And there's one more here that is also very significant. It was observed that bound, they keep saying viral particles, but they should be always saying viral-like particles. And you see in the section headings, they use the acronym VLP for viral-like particle because there was no virus used in this experiment. But here, it was also observed that bound viral-like particles detach after landing and diffuse away and even bind again elsewhere. So in other words, they don't bind the receptor of the cell, then go inside the cell and hijack the machinery and cause the cell to die, as we're told viruses do. They just bounce right off and go back into the void, away from the cell, and then later on they may bump into another cell because they're just I think what they're really describing here is a particle that's just randomly bouncing around and diffusing through a whole matrix of different extracellular proteins and other constituents, electrolytes and such in the extracellular solution. So in other words, not seeing anything meaningful. So I just want to go here to the discussion because this really tells you what the main focus of this paper is, which is that it's really trying to develop this technology, this tracking technology, which is combined or wed to this other imaging technology that they call 3D trim, because they feel this is a useful technique and they want to develop it and promote it. So what they write here in the highlighted portion, the skimming events observed here are, to our knowledge, the first of their kind and demonstrate how cell morphology can affect viral diffusion and how diffusion is inhibited at the cell surface. And we kind of talked about that. And the diffusion being inhibited at the cell surface means that as the particle got closer to the cell, it slowed down. And this could be because of, for example, a membrane potential, where there's a positive charge near the outside of the cell, and that could repel the particle away. So they didn't mention the possible electrostatic forces like that. Or it could be that the extracellular material is thicker around the outside of the cell, and so it was harder for the particle to diffuse. But they did, say that they observed this. And then going on, the distinct transport modes revealed here give insight into the different ways that VLPs, virus-like particles, interact with cellular protrusions. And I think that's a valid conclusion. It just doesn't tell us anything about nature or what really makes us sick. Finally, we demonstrated a rapid diffusion of single viral-like particles in a tightly packed epithelial layer, which to our knowledge has never before been reported. Well, so they did something unique, but too bad it doesn't tell us what we need to know about how nature works. Now, there's just one more section I want to review before I give my closing thoughts after we watch the video one more time. And this is in the methods section that I wanted to talk about how these viral-like particles are made, because it's quite an impressive endeavor. And so this highlighted portion kind of gives the meat of the matter and what it says, and it's very confusing to read. So I don't want you to read this here, but I did look up what these things are to verify, and I will tell you exactly what's going on here. So it says HEK293 T cells grown blah, blah, blah were transfected using a calcium phosphate-based protocol with either, and then it lists a whole bunch of things. So that means that they had cells in a cell culture. That's the HEK293T, and in a minute I'll tell you what those are. And they grew them in a cell culture, and then they added calcium phosphate, which must do something to the cells that they would take up genetic material. Then they mixed in a whole bunch of pieces of DNA, mostly in plasmids, which is a circular form of DNA that is found primarily in bacteria, but they use it for a lot of genetic experiments. And these plasmids are said to come from different viruses like HIV and like the VSVG that we talked about earlier, and even some others. And they put all these genetic material containing so-called viral genes, which came out of in silico genomics and metagenomics, just like I've taught before, and they put them in this cell culture. And this is the time to explain what these cells do. So here is the Wikipedia page for HEK293 cells, including 293T. And you can see right at the top is really as far as we need to go. It gives us all the important information. So their HEK stands for human embryonic kidney 293 cells, and they have some different names. And they are a specific, quote, immortalized cell line derived from spontaneously miscarried or aborted fetus, or human embryonic kidney cells grown on a tissue culture taken from a female fetus in 1973. So there's a lot to unpack there. But I think the key thing is that it's an immortalized cell line, which means it's been modified to grow indefinitely in the laboratory, unlike cells in nature, which die after a period of time undergoing senescence. So these are essentially kind of like cancer cells, and you should consider them genetically modified, and they don't resemble what would be in a real human kidney or even in the human kidney of a developing embryo. Now, the next paragraph says they've been widely used in cell biology research for many years because of their reliable growth and propensity for transfection. In other words, they would take up foreign genetic material and possibly express that, like in gene therapy. They are also used by biotechnology industry to produce therapeutic proteins and, quote, it should say pseudoviruses, but it says viruses for gene therapy. And in the next paragraph, it's a derivative of a human cell line that expresses a mutant version of the SV40T antigen. So that's key. So it's genetically modified, it takes up genetic material, and it expresses this SV40 antigen, which is associated with another made-up virus, SV40. So essentially what they would do is they would poison these cells in the culture. This is how they make the VLPs, or the virus-like particles. They culture these abnormal genetically modified cells. They add the chemicals to make them take up the genetic material. They give them all these plasmids with foreign genetic material. And then the cells undergo some kind of process where they put out particles, and some of them might even contain this genetic material. And that makes sense because this is essentially poison, and the cells want to get rid of it. So you have to, trick the cells into putting it in with the chemicals, and then they essentially try to get rid of it by putting it out through exocytosis into little particles. And those particles are what is exploited for this sort of, proxy research of doing in vivo experiments with these modified cell lines instead of actually studying nature. Okay, so let me go back and show you once again the video. And this time I will give some commentary as to what you are really seeing. It makes a trail going all through the tissue, and that purple blob now is essentially just the path that pseudovirus particle has been making in this extracellular matrix. Now in the background, you can see all these fluorescent yellow and red and orange and green substrate in the background. Now the large sort of spherical looking objects are essentially those human embryonic kidney cells in this monolayer, and then there's a lot of other stuff around them that we can't see because it doesn't stain. So you can continue to see this thing tracking around, but notice it's not really going to the cells, it's staying in the middle. But you might see it come near one of these cells, and they flipped it upside down to disorient us. But it certainly looks like we're flying through space, and now it seems to come, near to a cell, but it certainly doesn't exhibit any behavior that we would consider to be consistent with how we're told viruses infect cells and cause harm. So I'd just like to summarize that, first of all, this is not in any way a scientific experiment. And it's really important to state that up front to realize of what type of conclusions you may not be able to draw from this research. And the reason it's not a scientific experiment is because, one, there are no hypotheses stated that for testing. Secondly, there's no independent variable. Thirdly, there's no control experiment. In other words, they didn't use another type of particle, like let's say a synthetic lipid nanoparticle, with a fluorescent stain, but that doesn't contain any of that possible genetic material from the viruses, to see just how a regular man-made particle would behave in the same extracellular milieu. Because we very well could have seen that it behaved exactly the same, and in which case we could say that this is just how a generic particle of certain constituents behaves in a cell culture. But since there was no control experiment, we can't say that there's anything unique about these particular particles that they went to great lengths to manufacture that would be different from any other particle of the same size or similar materials. So at best, all we can say here is this is an observation rather than a scientific experiment and results. But this is not an observation from nature, which would be useful because, science is a way to help explain cause and effect relationships in nature. Like we observe a natural phenomenon, like someone getting sick, for example, or other natural phenomena like an earthquake. And then we have a hypothesis about what causes that thing we observe in nature, and then we can design experiments using science to test those hypotheses. So, if we might say that if I wear green sweaters, that means it's going to snow, we can easily design an experiment, right? Because snow is a real thing in nature. We've all seen it. We can design an experiment where I can purposely wear green sweaters and we see if it snows those days. And we'll quickly disprove my hypothesis that green sweaters are the cause of snow. But in this situation, we're not talking about something that's observed in nature because there's no natural elements in the experiment. The background cell culture is a genetically modified, commercially available cell culture that is not something we'd find in a real organism. And the viral-like particle is not at all found in nature. It's completely made in a laboratory with synthetic genes and proteins using this cell line. And I think the real purpose of this paper is really to develop this technology by some chemists. And I think the reason that they couched all of this research in terms of a virus is, one, because of the popularity and notoriety of virus research in the context of the pandemic, and two, because of the same kind of related reason, there's so much funding for this type of research based on viruses. So they could, use this to image a number of different types of things, right? But they might have specifically chosen, let's figure out how we can use this in the context of viruses so that we can get a research grant. And also Duke has a very, large biological and medical research institution. And so they would have the facilities there, for example, for the genetic plasmids to be constructed, on site at the university rather than having to try to do things like that themselves. So it's very important when you see a video like this or hear about it that you know some basic facts. For example, that we can't visualize things in a real life, in a living type of scenario, things that are that small as viruses. And there are many other videos actually that are just completely computer generated graphics, but this is not the case here. This is, it's computer generated in a way by calculating this very sophisticated laser tracking device system, but it's not a direct image. So essentially what we have here is something that doesn't explain anything about sickness or disease or cells or microbiology at all. It's a completely made up artificial experiment, and they made some observations about these artificial entities and can learn how they behave, but it has no relevance to our lives and our health. So once again, it's important to use your discernment to read these papers carefully, or find someone you trust who is capable of doing that and explaining to you, so that you don't get misled or fooled about this superstition, mythos, and outright fraud about imaginary viruses making us sick. Once again, I'm Andrew Kaufman, and this has been a Medicamentum Authentica special presentation. I hope it was helpful and maybe even a little entertaining. I'll see you next time.
Introducing one of nature's vital healing forces, from the alchemical science of spagyrics, made from the finest organic ingredients. Medicamentum Authentica brings to you spagyric cilantro. Studies have shown elevated levels of heavy metals like lead and mercury in people with various neurodegenerative diseases, such as dementia, Alzheimer's, and multiple sclerosis. It has been shown that cilantro can actually reverse the brain damage and remove these heavy metals from the body. The most powerful alchemical formulation of spagyric cilantro. All of nature was designed to harness a vital life force. Start your journey today and get the vital force of nature from every drop with Spagyric Cilantro by Andrew Kaufman MD.